Okay, good morning, dear students. So, we are starting our classes today. So, the topic will be the energetics and the regulation of glycolysis. Just a small recap of the previous thing. The process, what is the glycolysis? It is the breakdown of oxidative breakdown of glucose. The oxygen may or may not be present sometimes. So, total 10 steps are involved. First few steps, that is, first 5 steps are known as the preparatory phase, and the rest of the 5 is known as the payoff phase. So in the preparatory phase, the uh, the glycolysis actually starts. It will prepare, and in this stage, step number, as you remember, that step number 1 and step number 3, these are the two steps where the ATP is used. ATP energy is required to begin to start the glycolysis. And in the step number 7 and step number 10, these are known as the payoff phase. What is paying off? The ATP is paying off. Two ATPs are generated. And another thing is that from one molecule of glucose, by single glycolysis, two pyruvates are produced, because at the step number 5 there are production of two. Each of these two things will be produced from the step number 6, not 5. After 5, step number 6, two things will be produced. Two molecules will be produced. Two molecules will be produced. If it will start from the one molecule of glucose, after step number 5, there is production of two molecules of each of the substance. Two molecules of glyceraldehyde 3-phosphate, two molecules of 1,3-bisphosphoglycerate, two molecule of pyruvate, everything. So in the step number 1 and 3, these are the two steps where the ATP is required, plus these are the irreversible steps. Just look at there. Step number 1 and 3 are irreversible, and step number 7 and 10, these are the steps where the ATP is gained, is produced. These are also irreversible steps. All other rest of the steps are reversible, that is, it can be happen in both ways, forward and the reverse direction. Okay, so significance, simple thing, it is the metabolic pathway of one glucose into two pyruvates. It will produce into two pyruvates from one molecule of glucose. Two molecules of pyruvates are produced. It will give us the ATP energy. Not only the ATP energy, NADPH is also produced, which will give us energy if it will proceed through the further steps. It is important in the cell because glucose is the main source of fuel for the tissues in the body. It is an almost universal central pathway of glucose catabolism occurring in the cytoplasm of all the tissues. And most important thing, in case of RBCs which are having no mitochondria and no nucleus, so glycolysis is the only principal pathway of energy generation in the RBCs, because in case of RBCs, no TCA cycle and no electron transport chain is present there. So whatever energy it requires, it will comes through by the only way of glycolysis in case of human RBCs. So these are few of the significances of glycolysis. Uh, but listen, so what are the energetics of glycolysis? What are the energetics of glycolysis? So the energetics are, as we know that in case of step 1 and 3, 2 ATPs are required. So two ATPs are used, and in the step 7 and the 10, 4 ATPs are generated. Since they are having, each is having giving us two molecules of all the substances from starting from step number 6. So at the step number 7 and step number 10, 2 ATPs, 2 into 2, that is total 4 ATPs are generated. So what is the sum total? The sum total is that when the condition is anaerobic, that is absence of oxygen, step number 7 and step number 10 will give us total 4 ATPs. And in case of the preparatory phase, in step number 1 and 3, it is undergone through the wastage or uses of 2 ATPs. So 4 minus 2, the sum total is the 2 ATPs, or the net gain per molecule of glucose is 2 ATPs. But if it is going on through the aerobic condition, oxygen is present, That means the NADH or the NADPH which is produced in step number, which step number? Just remember, NADH is produced in step number 6 there. Just look at from glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate. NADH is produced in the step number 6. So or NAD is produced, NAD plus to NADH plus H plus. When they are produced, if the condition is aerobic, that means if the mitochondrial electron transport chain is there, when this NADH will pass through this mitochondrial electron transport chain, mitochondrial enzyme, malate shuttle or phosphoglycerate shuttle, whatever, they will give us further energy. Two types of shuttle systems are there in the mitochondria. One is the malate shuttle. If that NADH which is produced in the step number 6 during the aerobic condition will pass through the malate shuttle of mitochondria electron transport chain, it will give us 5 ATP, or if it will pass through the glycerophosphate shuttle, it will give us 3 ATPs. So what is the sum total of our ATP production in case of aerobic condition? 2 to 4, that is 2 plus 2 4 ATP in step number 7 and 10, plus either 5 ATP from NADH in the step number 6 if it will pass through the mitochondrial electron transport chain. It will be 5 ATP if malate shuttle. Just look at the table, this portion. It will be 5 ATP if it will be using the malate shuttle, or it may be 3 ATP if it will be using the glycerophosphate shuttle of mitochondrial electron transport chain. So the total is the 9 or the 7 ATPs when oxygen is present. These are the, we are gaining these ATPs. These are the payoff phase. In case of preparatory phase, 2 ATPs are wasted. So the net gain per molecule of glucose 9 minus 2 or 7 minus 2 is 7 or 5 ATP. So the total thing is that when the oxygen is absent from one molecule of glucose, we are getting net gain. I repeat again, net gain of 2 molecules of ATP, 2 ATPs. If the oxygen is present, if the condition is aerobic, and if the NADH will pass through the mitochondrial electron transport chain, at that condition from per molecule of glucose, the net gain will be 7 or 5 ATPs. So that is the energetics. Same thing is explained here when the condition is anaerobic or when the condition is aerobic. Next is the inhibitors of glycolysis. What are the inhibitors of glycolysis? Number of inhibitors may be present there. Inhibitor means actually they will inhibit the glycolysis in different steps. They will inhibit the action of the enzyme. Any step. Say for example, if you consider the step number 1 or step number 2, where they are applicable, they causes the inhibition of the enzyme which are responsible for that step. So these are the function of inhibitors. Number of inhibitors are there. Say for example, 3-bromopyruvate is there, iodoacetate is there. You will go through, if you will go through some of the literatures, you will get some fluoride blocks. Blockers are there, fluoride blockers, which will block glycolysis by inhibiting the magnesium plus 2 dependent enzyme enolase. The enolase enzyme can be inhibited, which is a magnesium dependent enzyme. That enolase enzyme can be inhibited by a fluoride blocker. So that fluoride is acting as a inhibitor of glycolysis in case of enolase, or some other inhibitors like iodoacetate or iodoacetamide, which can bind covalently to and alkylate the SH group or the sulfhydryl group of glyceraldehyde 3-phosphate dehydrogenase and thereby inhibit the enzyme. So the isoacetate or the iodoacetate or iodoacetamide, these are the two inhibitors which can bind covalently and alkylate the sulfhydryl group of the one enzyme. Thereby it will reduce or it will stop the activity of the enzyme. How they will stop? By covalently binding and alkylating the sulfhydryl group. The sulfur and the hydrogen group of enzyme. What is the enzyme? The enzyme is glyceraldehyde 3-phosphate dehydrogenase. So they have the ability to inhibit the enzyme glyceraldehyde 3-phosphate dehydrogenase. What are the those name of those blockers or inhibitors? Those are iodoacetate or iodoacetamide. These are the some of the blockers also given there. Oxamate is there, TTFA, FCCP, oligomycin. There also their full forms, full names are given in the below of the table. Oligomycin, the percentage of inhibition and inhibition percent, everything is not. These are not important for your exam, but also just to know what are the just you have to know the what are the inhibitors and how they will inhibit the glycolysis at which step, or they will act on which enzyme. Okay, next is the regulation of glycolysis. Now the how the glycolysis is being regulated. So see, there are two conditions actually. When we are having the sufficient amount of glucose or sufficient amount of carbohydrate in our body. Second one, when we are not taking any food, any meal, or somebody is in fasting condition. So the general idea is that when carbohydrate is present in our body, or we are taking some carbohydrate meal, the normal phenomenon is that blood glucose level increases, which stimulates the insulin secretion. This insulin secretion increases the synthesis of glucokinase, phosphofructokinase and pyruvate kinase. Plus insulin and glucagon that enhances the uptake of that glucose, that I already told in last classes, that the glucose is circulating throughout the blood, but in the blood glycolysis is not going on. That glucose must be entered inside the cell and that will reach the cytoplasm. So they will enter, they will reach the cytoplasm due to the action of these hormones insulin, glucagon. Plus there are specific transporters named as GLUT transporters, which are present in the plasma membrane of all the cells, through which these glucose can be entered inside the cell. So that is when the carbohydrate meal we are taking. When a person is in the fasting, since the person is already in the fasting, his or her blood glucose level falls. That inhibits the insulin secretion and stimulates the glucocorticoid secretion. That causes or increases the synthesis of the 4 enzymes that reverses glycolysis, stimulates gluconeogenesis. Gluconeogenesis is a process where glucose are produced by some non-carbohydrate sources. From that sources glucose can be produced, that is gluconeogenesis. Somewhere it is termed as new glucogenesis, same thing. So these are the general idea. Now if you enter, I want to remind you that if you remind the steps, how many steps are there? 10 steps are there. First 5 is the preparatory phase, where step number 1 and step number 3 are irreversible, where we need 1 that is 2 ATPs to start, and from step number 6 to 10 these are the payoff phase. At step number 6 we are getting some NADH. In step number 7, how many NADH? 2. And in step number 7 and step number 10 we are saving, preserving 2 plus 2, that is 4 molecules of ATP. That is the picture. Now how, so and this step number 7 and the 10 are irreversible. Rest all other steps are reversible in both sides, both way direction. Now so there are some of the enzymes which are responsible for the regulation of glycolysis. So regulation means if you can regulate the enzymes, you can control the steps of glycolysis. You can even stop the glycolysis. You can even enhance the rate of glycolysis. You can increase or you can decrease the rate of glycolysis. Even you can stop or start the glycolysis. So that is the regulation. So that regulation is actually based on regulating the enzymes which are controlling the glycolysis. So at which steps? What are the enzymes which are involved in the regulation of glycolysis? That is the topic of our discussion. So first enzyme which is involved in step number 1, which is also a irreversible step, which is also the first step of payoff phase, which is also the first step where we require one molecule of ATP to being used, and the enzyme which is involved is the hexokinase. Second enzyme, which is phosphofructokinase 1. Where this phosphofructokinase 1 is required? This phosphofructokinase uh, is required in the step that is from fructose 6-phosphate to the fructose 1,6-bisphosphate. That is the step number 3. This step number 3 is again a irreversible step. This step number 3 is the second step of the preparatory phase, and this is the second step where we are going to using another ATP molecule. Another ATP is wasted. That is the phosphofructokinase 1. Step number 3. Next enzyme, that is the pyruvate kinase. Where the pyruvate kinase is required? The pyruvate kinase is required at the last step for the production of phosphoenol pyruvate from the phosphoenol pyruvate. That is step number 10. This is again a irreversible step, and this is the step where we are gaining, where we are acquiring, or where we are getting two molecules of ATP. That is the pyruvate kinase. Fourth enzyme is involved. This is not directly related with the glycolysis which is going on in body tissues, but this is related to the glycolysis which is uh, going on in the muscles. That means this is involved with the muscle glycolysis, which is started from the muscle glycogen. As we know that the glucoses are stored in the muscle, okay, but that will be stored in the form of glycogen. So when there is energy requirement uh, in the muscle, that glycogen is converted into glucose, and that glycogen will enter to the glycolysis. That stage where the uh, initiation of the glycolysis of muscle glycogen is necessary. One enzyme which is responsible for that step, I repeat, when there is the necessity of initiation of glycolysis of muscle glycogen, the enzyme responsible is glycogen phosphorylase. So glycogen phosphorylase is our fourth rate limiting enzyme. This you can known as the, you can call as rate limiting enzyme, which is limiting the rate of the that process. It may be glycolysis, it may be some other process. So these are the four enzymes. So first is the hexokinase. What is hexokinase? The first enzyme. So it is allosterically inhibited by its product glucose 6-phosphate. What is allosteric inhibition? You are having this enzyme and all these things, I think, in your first semester, if you can remember. What is the allosteric enzyme? Allosteric means other site. That means, as we know that the enzyme has a specific site which is known as the active site. When a substrate will bind to the active site, it will fit with the active site, and that substrate will work on the enzyme and on the product, and that substrate, and it will make the product. Okay. But other than this active site, some of the enzymes are having some other sites which are known as the allosteric sites. So what are their functions? Sometimes some metallic ions, sometimes some vitamins, or some other maybe some other proteins will bind to that site. Second, this is the second site, not the active site. They bind at the second site, and this can modulate, this can regulate the activity of the enzyme. So that is the allosteric enzyme. So in a nutshell, in a simple way, allosteric enzyme is an enzyme which is having a specific site where the binding of some other substances can regulate the activity of that enzyme. This is not the active site. This is not the site where that enzyme will bind with its substrate. But this is a site which is other than the active site, where the binding of some other substance, where the binding of some other proteins, or the some metallic ions like magnesium Mg plus 2 or Mn, or some other vitamins, they can activate or they can modulate the activity on enzymes. Okay, so all the enzymes which are involved in this glycolysis are actually, they are all are having some allosteric site. So what is the allosteric site? This is the second site, not the first site. First site is the active site where the enzyme will bind with the substrate. Okay, but all of these enzymes, most of the enzymes are having a second site. that second site is known as the allosteric site. So allosteric site is actually a modulator site, is actually a regulator site for that enzyme. Where I am talking about the allosteric site, that is the second site. Don't get confused. So the allosteric site is a site where the binding of some other substance, it may be a vitamin, it may be a protein, it may be some metallic ions, binding or non-binding at the second site, it can regulate the activity of the enzyme. It can sometimes increases the activity of the enzyme, it can sometimes decreases, or it can sometimes stop the activity of the enzyme. So that is the allosteric site, that is the allosteric enzyme, and that is the allosteric regulation. Okay, now come to our this topic. So hexokinase, so it is a allosterically inhibited by its product. See, so what is the product of this hexokinase? Where it is used in the first step, the steps are glucose to glucose 6 phosphate. The enzyme is hexokinase. So the enzyme is hexokinase, substrate is glucose. What is the product? Product is the glucose 6 phosphate. Now what is written that this hexokinase, which is having a second site, allosteric site, the its product is the glucose 6 phosphate. Now this glucose 6 phosphate can bind to its second site or allosteric site, and by binding it can regulate, it can allosterically changes the activity of the enzyme. What type it will change, whether it will inhibit or activate? It is written that in this case the glucose 6 phosphate allosterically inhibits the activity of hexokinase. In the liver, another form of hexokinase is there, that is known as glucokinase. So in the liver, the glucokinase is inhibited by the fructose 6 phosphate. In the liver, its product is the fructose 6 phosphate. Now this fructose 6 phosphate allosterically inhibit the glucokinase in the liver. So in case of other cells, hexokinase is present. In case of the liver cells, the glucokinase is present. All are the same types of enzymes. These are known as the isoenzymes. If you already know the idea of isoenzymes. What are the isoenzymes? Isoenzymes are different types of enzyme, different subtypes of enzyme. These are the isoenzymes. So the hexokinase or the glucokinase, all are isoenzymes. Glucokinase are present in the liver and hexokinase in other tissues. Okay, so hexokinase reaction is metabolically irreversible. Yes, it is irreversible as it is shown by the single arrow in the first step. We know glucose 6 phosphate is the product. Glucose 6 phosphate level increases when the glycolysis is inhibited at sites further along in the pathway. Actually, total glycolysis is not inhibited when the concentration of glucose 6 phosphate increases. See, just think about the condition, think about the logic. First step, glucose, we are having one molecule of glucose, that is the substrate. The enzyme is hexokinase, the product is glucose 6 phosphate. Now it is going on. When there is the excess concentration of glucose 6 phosphate, so there is no further necessity of breakdown of this glucose into glucose 6 phosphate. So now the allosterically that hexokinase will be inhibited. That means cell is sending a signal that I am having sufficient amount of glucose 6 phosphate, no need to further conversion of glucose into glucose 6 phosphate. That causes the reversible allosteric inhibition of the hexokinase. See, it is written that glucose 6 phosphate inhibits hexokinase isozyme, and the same form of a enzyme. So this hexokinase is having hexokinase or glucokinase, whatever the name, they are having 4 types, 1, 2, 3 and 4. The glucokinase is a hexokinase actually, which is the type 4 of glucokinase. So type 4 glucokinase, which is present in the liver, is termed as the glucokinase, and all the other 3 types 1, 2 and 3 are present in the other body cells. So hexokinase, which is allosterically inhibited by its product when there is the excess amount of product. So that is the regulation of hexokinase. Next, you see, it is also given that, just look at the figure. The glucose is transported inside the cell from the blood because of presence of a transporter, that is GLUT, G-L-U-T, GLUT2 in this case. GLUT number of other GLUTs are GLUT3, GLUT4, all these are there. In this case, GLUT2, which is present in the plasma membrane, this helps to transport glucose from the blood to inside the cell. Now this causes the conversion of glucose to glucose 6-phosphate or fructose 6-phosphate. Now this glucose 6-phosphate and this or the fructose 6-phosphate, they are having negative allosteric feedback effect on hexokinase 4 or hexokinase 1. Hexokinase 4 in case of liver cell, that is glucokinase, or hexokinase 1, 2 or 3 in case of other cells. So glucose 6, that is the product of hexokinase, causes the reversible allosteric inhibition, not the activation, inhibition of hexokinase, that is showing. Plus the hexokinase which is produced from the nucleus, since as it is the protein, it will must undergo through the replication, transcription, translation. After translation in the nucleus, that will come out and it will forming a hexokinase enzyme. So that hexokinase can be further regulated by some regulatory proteins which are present in the nucleus. That is the regulation of hexokinase. Next enzyme we are having, that is the phosphofructokinase 1. Phosphofructokinase or the phosphofructokinase 1. So can you remember where the phosphofructokinase 1 is involved? Yes, you are right. The phosphofructokinase is involved in step number 3. That is again the irreversible step. So phosphofructokinase mediates the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate, which requires magnesium ion plus 1 ATP energy is needed. This is again a allosteric enzyme. It is activated by ADP and the AMP. So just let us enter the main step. So phosphofructokinase is the principal rate limiting enzyme of glycolysis, which is again a allosteric enzyme. That means it is having a second site where the binding of something can modulate its activity some way. What is the something in some way? We are going to discuss next. So the regulatory factors of phosphofructokinase 1 are: one is the physiological concentration of ATP, one is the citrate, one is the fructose 2,6-bisphosphate. So in the resting muscle, the concentration of ATP inhibits the phosphofructokinase 1 allosterically. When the ATP binds with the allosteric site of phosphofructokinase, it inhibits the PFK1 activity, where in case of the resting muscle, and thereby it reduces the substrate affinity for fructose 6-phosphate. So it inhibits and it reduces the activity of fructose 6-phosphate. So fructose 1,6-biphosphates are not producing. A rise in the ATP ratio decreases the activity of phosphofructokinase 1. Rather, a fall in the ATP or ATP AMP or ATP ADP, whatever form of phosphates are there, it may be triphosphate, monophosphate, diphosphate. A rise in the ATP causes the inhibition of the PFK1, or a fall in the ATP level causes the activation of the PFK1 activity, which correspondingly changes the rate of the glycolysis. That means if the ATP concentration is high, the phosphofructokinase 1 is allosterically inhibited. If the ATP concentration is low, so ATP concentration low means what? You require more ATP. So if you want to acquire more ATP, that glycolysis must be going on. The glycolysis should be proceed. That means the step number 3 should be overcome. Then you have to enter 4, 5, 6, 7, 8, 9, 10. So if the ATP concentration is low, that allosterically activates the PFK1. Therefore, step number 3 and the next consecutive steps are going on. That is the ATP concentration on PFK1. Second one is the citrate. What is citrate? Citrate is not directly involved with the glycolysis, but it is involved with the TCA cycle. Citrate is a TCA intermediate. Okay, citrate is the TCA intermediate. This citrate also acts allosterically. That means this citrate can also bind at the second site of PFK1. So citrate, which is a TCA cycle intermediate, this can act allosterically and augments the negative allosteric effect of ATP on the PFK1. That is, it augments the action of ATP on the PFK1. What is the action of high concentration of ATP, high concentration of ATP allosterically inhibits the PFK1. These TCA cycle intermediates augments this negative allosteric effect of ATP on the PFK1. That means this supports citrate supports. Third one is the fructose 2,6 bisphosphate. So fructose 2,6 bisphosphate acts as the principal allosteric activator of the phosphofructokinase 1. That means it activates the PFK1. Now it is not inhibiting fructose 2,6 bisphosphate. Now activating the PFK1 by allosterically, and it counteracts the negative effect of ATP. The whatever the ATP is doing in the just see in the previous two paragraph, whatever ATP is doing, what ATP is doing, it inhibits the PFK1. Now this case fructose 2,6 bisphosphate reverses the action of ATP and it activates the PFK1 allosterically. So fructose 2,6 bisphosphate activates the PFK1, citrate and ATP concentration inhibits the PFK1 allosterically. Okay. So next enzyme is we are having, that is the pyruvate kinase, simple one. Where the pyruvate kinase is necessary? The pyruvate kinase is necessary at the 10th or the last step where we are getting two ATPs which mediates the conversion of phosphoenol pyruvate to enol pyruvate or pyruvate. So the role of pyruvate kinase is that when sufficient energy supply is ensured in the cell by high cellular concentration of free fatty acid or the acetyl CoA or the ATP, this inhibits both the hepatic and the muscular pyruvate kinase isozyme allosterically. Again allosterically, when the high cellular concentration of free fatty acids or the energy or the acetyl CoA or the ATP is present, this allosterically inhibit the pyruvate kinase where in the hepatic, that we hepatic means liver, in the liver cell as well as in the muscle cell. In both the sides, these substances allosterically inhibit the pyruvate kinase enzyme. But the rise in the cellular fructose 1,6 bisphosphate, second view is that when there is a increase in the cellular fructose 1,6 bisphosphate, this fructose 1,6 bisphosphate causes the reverse thing, that is this allosterically activates the pyruvate kinase. All these substances, acetyl CoA, fatty acid, ATP, all these substances causes the inhibition of hepatic and the muscular pyruvate kinase, but in this case the fructose 1,6 bisphosphate causes allosterically activation of this pyruvate kinase. So that is the regulation of pyruvate kinase. The last enzyme, which is, yeah, that is the line. So the last enzyme is the glycogen phosphorylase. Where the glycogen phosphorylase is involved, it is not directly related with the glycolysis, but it is important in the muscle. As we know that the most of the glucose are preserved in the form of glycogen. So when the energy requirement is there, the glycolysis of muscle glycogen requires the enzyme glycogen phosphorylase. Now this glycogen phosphorylase exists in two forms. One is the phosphorylase B and phosphorylase A. Phosphorylase A is more active and having phosphoserine residues. Phosphorylase B is inactive and having no phosphate group on its serine residues. Some of the charts are given how the muscle glycogen phosphorylase regulates. These are the overall reaction of regulation of the glycolysis. You can add this chart also, the overall reaction. All the enzymes are showing in which step, where and how they are regulating the glycolysis. Allosterically means all are actually the allosteric enzyme which is having a second site, which is the allosteric site, where the binding of some other substance can modify the activity of the enzyme. That substance may be its product, maybe some metallic ion, maybe some other hormones or some other vitamins maybe. So that's all for today's class.